Okay, well, here we go. Oh, finished drinking that already. That's unfortunate. I want to go over this preprint paper. So it has not been peer reviewed yet. It's in the process of peer review, and it is a very long, like more than 3 decade long longitudinal study of a group of 10 people to understand how the four normal human coronaviruses, how what is the reinfection rate for them? So there are many mechanisms by which you can become reinfected by a disease. And what I really should say, it's not a question of if you can get reinfected by a disease, because for every disease, you are probably going to get reinfected with them. Or there's a good probability that you will get reinfected by any disease, even if you're immune. But the whole point of immunity is that you do not experience some of the pathology that is associated with the reinfection. So you are able to have antibodies in there that are able to slow down the duplication of the virus or whatever pathogen you end up consuming or getting infected with. And then on top of that, the the immune cells that are expressing the antigens, or I should say, are expressing the antibodies on their surfaces for those particular pathogens, they end up duplicating very quickly in response to those pathogens, and therefore you're able to quickly uh, take hold over a reinfection event, so that you do not end up having pathology associated with it. Now, as I said yesterday, in regards to the antibody-dependent enhancement, which is seen in quite a few diseases, um, the antibodies can actually work against you and make the second and subsequent events far more deadly than the first. So, with any type of reinfection, there are multiple courses that can happen. Either you have absolutely no pathology, or you can have the exact same pathology, or you could have a worse, or you could have a far worse pathology associated with the subsequent contractions of those diseases. So, um, what I want to do right now is discuss how for pretty much all coronaviruses, or I should say all human coronaviruses, you can get reinfected with all of them, and you can get reinfected and display similar pathology every single time you get infected. And this longitudinal study, although not yet peer reviewed, supports those claims. But then this also works in conjunction with the other studies on SARS and MERS, where we definitely do see antibody-dependent enhancement in model organisms and in model cell cultures. So we have plenty of reason to believe that not only will you be able to be infected a second, third, fourth time, but we also have reason to believe that you are going to display pathology each and every one of those times, or not, I shouldn't say each and every one of those times, but you are likely to experience pathology if you do end up contracting it, especially when the antibody titers or the antibody concentrations are within certain ranges. Because as yesterday's video explained, for the known coronaviruses, we see this happening. We see if you have certain levels of antibodies, if they're just enough to be sub-neutralizing, so you don't have so much of them that the virus particles are completely coated and rendered useless, it can actually facilitate the infection of certain immune cells. So these antibodies will coat partially the virus, and then the immune cells will be attracted to those antibodies because they have the Fc receptors on them. And if everything lines up correctly, it is very possible for a virus, even though you have immunity, theoretical immunity to this virus, it ends up getting incorporated into the immune cells, and ends up duplicating within the immune cell. So you've got to be very vigilant about your antibody levels. And this particular paper brings to our attention how for the coronaviruses we do tend to very quickly lower our antibody levels for them. Now, why would our body want to lower our antibody levels? Well, for one, it takes a lot of energy to produce these antibodies. So we will have our high expression of antibodies for a period of time, and then we lower them to sort of a basal level. Um, and what is the basal level? Well, that depends on what antigen you're talking about. So oftentimes, especially if you're getting vaccines, sometimes you need booster vaccines because what will happen is you will initiate an immune response and you'll get a lot of antibodies produced from that vaccine, but then slowly it decreases and then it goes down to a very low level. But the second time you get infected, or I shouldn't say infected, but the second time you get the booster shot, it ends up, or the first booster shot, I should say, you can create a lot more antibodies than the first time, and then the new basal level is a little bit higher than the first time. And we would hope that the higher basal level is protective. Okay, but when it comes to the known human coronaviruses, the antibody levels for them get below their basal level is below the amount for getting reinfected with the pathology. So you will still have antibodies, or you might actually not have any of the antibodies left, but they're so low that you are able to get reinfected and show pathology when you get reinfected with these other four coronaviruses that spread around the population every single year, or at all times, I should say. So, but, and I also want to raise a question: why else would you want to lower your antibody responses? Well, uh, there are some problems with expressing antibodies, especially if they are cross-reacting with some of your own peptides and peptide hormones. So these would be forms of autoimmune diseases. So this is why it's not always good to be expressing a lot of antibodies because those antibodies could be damaging your own body. So lower the antibodies when you don't need them and have them at a very low basal rate. Okay, because especially if you have a lot of autoimmune reactions, it could be very difficult to ramp down that antibody response because your immune cells are always getting activated to produce more antibodies and to duplicate even more. And so that could perpetuate an auto antibody attack on your own system. And we do know that this is a strategy that is employed by both influenza and coronaviruses. Specifically, the one we know about is the targeting of the corticotropin hormone, also known as ATCH, and this hormone is used for the release of cortisol, and I assume other glucosteroids as well. But this can influence your hormone levels, which is not necessarily the best thing you want to do, or the best thing, or anything that you want to happen to yourself, especially when you have other diseases that you got to fight. So, what I'm going to do now is go over this preprint paper that was a longitudinal study of 10 people in, I believe, the Netherlands. And it was basically going over their antibody levels and how low did their antibody levels have to get before they were susceptible to the same type of the same strain. I shouldn't say same strain. I should say the same species of coronavirus. So these people, even though there are four different coronaviruses or four different normal human coronaviruses that are always circling around, um, they're, how should I say this? Even though there are only four that are always, I shouldn't say only four, even though there are four circling around, you can get infected by each of them multiple times throughout your life. And the amount of time it takes in between each reinfection doesn't seem to be very long. Okay, so this is not necessarily boding well for the other more serious coronaviruses. And uh, MERS and the original SARS, we got under control and basically eradicated. Uh, we functionally eradicated it from the human population. Luckily, MERS was not as contagious from person to person, and but this current pandemic, this is the first pathogenic, highly pathogenic SARS that is spreading, that is pandemic in the modern era. Now, this doesn't say whether or not the other human coronaviruses were, they might have been much more pathogenic when they first crossed over into humans, or they might have been very pathogenic at a period of time while they were endemic to the human population. But in the current modern era, all four normal human coronaviruses are pretty benign. So one of my worries now is that we are now going through a pandemic where we are introducing ourselves to a highly pathogenic coronavirus strain, and at least the first run throughout the human population, it is going to cause a severe change in human health and public health. So let's begin. So this is the title, and it's on medRxiv, which is a preprint server. And it's the Human Coronavirus Reinfection Dynamics Lessons for SARS-CoV-2. And what I find amazing is that this was a longitudinal study that was 3 decades, and they decided to rush it out when this became a pandemic. So, abstract. In the current SARS-CoV-2 pandemic, a key unsolved question is the quality and duration of acquired immunity in recovered individuals. This is crucial to solve. However, SARS-CoV-2 has circulated for under 5 months, precluding a direct study. We therefore monitored 10 subjects over a time span of 35 years, providing a total of 2,473 follow-up person months, and determined A, their antibody levels following infection by any of the 4 seasonal human coronaviruses, and B, the time period after which reinfection by the same virus can occur. An alarmingly short duration of protective immunity to coronaviruses was found by both analyses. We saw frequently reinfections at 12 months post-infection and substantial reduction in antibody levels as soon as 6 months post-infection. Okay. So the main text. SARS-CoV-2 is a novel coronavirus responsible for an ongoing pandemic. Its rapid transmission is most probably caused by the fact that the virus entered a grossly naive, thus highly susceptible human population, combined with the capacity of the virus to transmit during the asymptomatic phase of infection. Since no pharmaceutical interventions are universally available or applied, current policies to limit the spread of SARS-CoV-2 revolve around the containment, social distancing, and the assumption that recovered patients develop protective immunity. So right now we are challenging this particular assumption. So a lot of our models regarding immunity and herd immunity and the rate of epidemic spread, we use the SIR model. So the susceptible, infected, and the we can say recovered or removed. So we use the SIR model, which is an imperfect model because it is a highly what would the word be, idealistic model. So one of the problems in that is that the population is always changing, so you're always birthing new people into the population. So herd immunity is a lie. So I shouldn't say it's a lie, but to achieve herd immunity and then assume that herd immunity means that the disease is eradicated, that is not the case. That is absolutely a false premise. When you reach herd immunity, that means that whatever pathogen it is no longer spreading uh, exponentially. It basically means that the R value is 1, or close to 1 naturally. So, but it does not mean that, um, how do I phrase that? It does not mean that herd immunity or that protective immunity is going to last, because new people are born into the world all the time. And so rather than burning through the population, it's more like a smolder. And then it'll smolder until more and more people are born into the world that are naive to this virus, which dilutes the immunity until it goes below herd immunity again. And once it goes below herd immunity for a significant portion of time, then eventually you can have another wildfire of the disease spreading very quickly again. And then, and that's one of the assumptions with the SIR model that isn't exactly perfect. The other one is that once you are in the R, once you are removed, or once you are recovered, that you have protective immunity. And we know that for all of the known coronaviruses, all the known human, I should say, normal human coronaviruses, so the 4 that go around all the time, that is not the case. You can get reinfected from them and you can show pathology when you get reinfected again. Now, is that due to your antibodies never being imperfect? Is it due to your antibody levels going down? Or is it due to the mutation rate of the coronaviruses? Because once it's endemic in the human population, it's had plenty of time to go around the entire population and accrue immunologically relevant mutations, which allows it to evade antibodies. So that could be another option that allows viruses to be more infectious. Um, we haven't, we don't know that exactly, but this paper is trying to say that perhaps one of the factors in why you can get infected with coronaviruses a lot is because the antibody levels drop precipitously, so more so than average. So let's continue. It is still unclear whether protective immunity is indeed induced after infection and for how long. The duration of protection will impact not only the overall course of the current pandemic, but also the post-pandemic period. To date, no concrete evidence of reinfection by SARS-CoV-2 is available, nor is any example of reinfection by SARS-CoV-1 or MERS-CoV. So, yes, we cannot do these experiments on people. So with the first SARS and with MERS, I think you'll be very hard pressed to find a survivor of one of these diseases that would be willing to be injected with the virus again. That's just a fact. I mean, not a fact. I mean, that's a very reasonable expectation. And nor if I was a researcher would I even want to ask a survivor to be infected again with these highly pathogenic diseases. Okay, so to date, no concrete evidence of reinfection by SARS-CoV-2 is available, nor is an example of reinfection by SARS-CoV-1. But remember, what we're saying here is that SARS-CoV-2, how to phrase that, has not been circulating for that long. So we would assume that all of the survivors of SARS-CoV-2 still, especially if they had a serious disease, would be still be producing high levels of antibodies and protective antibodies, which would theoretically protect them from a recurring infection. Anyways, yet it is likely influenced by the recent emergence of SARS-CoV and the limited scale of the SARS-CoV-1 and MERS-CoV epidemics. The limited available data on potential protective immunity against the coronavirus reinfection is derived from one experimental infection study in volunteers, from which it is generally assumed that reinfection by coronaviruses can occur. So this must have been an experiment with the normal human coronaviruses, not the SARS or MERS, from which it is generally assumed that reinfection by coronaviruses can occur. Natural coronavirus infections and susceptibility to reinfection have not been investigated thus far. If they occur, reinfections will probably be dictated by two variables: exposure to the virus and the quality of sustained immunity. Okay, continuing. Even though it is not possible to investigate SARS-CoV-2 reinfections yet, the seasonal coronaviruses may serve as a reliable model. There are four species of seasonal coronaviruses: the HCoV, NL63, 229E, OC43, and HKU1, and all are associated with mostly mild respiratory tract infections. However, aside from being etiological agents of common cold, the four viruses are biologically dissimilar. Two belong to the genus alpha coronaviruses, and two to the genus beta coronaviruses. The viruses use characteristic receptor molecules to enter a target cell, and based on receptor distribution, they do not all enter the same epithelial cell type in the lungs. So, depending on what receptor, that determines what type of tissues you are able to interact with. Continuing, given this variability, the seasonal coronaviruses are the most representative virus group from which to conclude general coronavirus characteristics, particularly common denominators like host protective immunity and susceptibility to reinfection. Since most people experience their first seasonal coronavirus infection during early childhood, reinfections later in life can be investigated. Okay, so what they're saying here is that since the four other human coronaviruses are so genetically dissimilar, we can safely assume that they are somewhat of a representative sample of what general coronaviruses do in the human population. Um, Granted, you could also go further and say they're doing something that most viruses do. Most viruses can reinfect you, but they might not be pathogenic the second time they reinfect you. Um, but when it comes to these common cold coronaviruses, it sounds like the infection is symptomatic to a degree, and they are causing the mild respiratory tract infections. I believe one of those is also an enteric coronavirus, and that it is able to cause intestinal problems, but I would have to look back on that to see if it is one of those four. Continuing, the aim of the study was to investigate the time period between the coronavirus reinfections and the dynamics of antibody decline following infection. These parameters were assessed by measuring the immune response to each individual seasonal coronavirus over an extended period. We followed healthy subjects for multiple decades at regular intervals. As IgG, which is a type of antibody, these are the long-term antibodies, or one of the long-term antibodies, as IgG levels only increased after successful infection remained and importantly remained constant after unsuccessful viral challenge. Increased antibody levels identify an infection. We use the carboxy terminal of the nucleocapsid protein as the target antigen in ELISA, as this protein is immunogenic, relatively low in interspecies conservation, yet high in intraspecies conservation when compared to the spike protein. So let me just reread this again. Increase. Okay, so rather than dealing with the spike protein, which tends to mutate a lot more, they decided to use the nucleocapsid protein to measure the antibodies for that instead. Because, let's say, your coronavirus is mutating significantly, um, then the antibodies might not be cross-reactive. So then one could start making the argument that the coronaviruses are just mutating very quickly, and that's why you're getting reinfected, rather than just the low concentrations of antibodies causing the reinfection. Continuing, frequency of infections and reinfections from a prospective cohort study following adult males. 10 subjects were randomly selected. Follow-up of subjects started in 1985, and besides a gap in follow-up between 1997 and 2003, continued until 2020 at regular intervals, every 3 months prior to 1989, and every 6 months afterwards. The cumulative period at which subjects were continuously followed totaled more than 200 person years. At start of the study, subjects aged ranged from 27 to 40 years. By the end of the follow-up, subjects were 49 to 66 years old. The study was approved by the Medical Ethics Committee of the Amsterdam University Medical Center of the University of Amsterdam and Netherlands. So this is probably an effective study because it's longitudinal, it's dealing with the same 10 people, and there is regular testing with just a small gap, and therefore we can probably learn a significant amount from this type of study. Now, one would probably want the sample size to be much larger, right? But it's very hard to get both a large sample size and do a longitudinal study where you're following that large sample size throughout their lives. It's just difficult to have that type of continuity. Continuing, infections by seasonal coronaviruses were defined as a 1.4-fold increase in antibody levels occurring between 2 visits. And additionally, the standard deviations of the mean at both visits not overlapping. A total of 132 events, ranging from 3 to 22 per subject, met these criteria and were thus considered infections. Oh, so they're saying is, and this might be one of my problems with this particular paper, but so I'll raise it. They're saying if your antibody levels increased during the time by a 1.4 fold increase between 2 visits, and additionally the standard deviation of the mean at both visits were not overlapping. So basically, I assume they're taking multiple samples of antibodies at each visit, and then they make sure they plot one graph, and then they plot another graph, the follow-up, and if those 2 curves are significantly different and at least a 1.4 fold increase in antibody levels, then they assume that you were infected in that time frame. Now, what I would say is that there might be other events that can cause your antibody levels to increase significantly, such as especially if you have an autoimmune dysregulation, you might actually start producing a lot of antibodies. Like you might produce more antibodies than you normally would. So that would be something that might increase your antibody levels or concentrations. But let's assume these people know what they're doing, and they've obviously been doing this research for a long time. So I'm putting some stock in their expertise right now. But they're saying that is the criteria for a reinfection. Continuing. And additionally, the standard deviations and the incidence rates of seasonal coronavirus reinfections per 100 person years of follow-up was 15.3, 20.1, 16.4, and 6.3 for the HCOV, NL63, 229E, OC43, and HKU1 respectively. So let me just double check that. So out of 100 person years, out of 100 person years, you would expect 15 0.3 infections for NL63. For 100 person years, you'd expect 21 infections of the 229E, and so on and so forth. So what they're saying is, if you live to be, let's say you live to be 100, you're probably going to have about uh, this many infections on average. But granted, they weren't talking about one person, they're talking about 10 people. So it's more like out of every 10 years, for 10 people, you're going to have about that many infections. So, yes, that would be about one infection, 1.5 infections every decade per person. Wait, let me. Yeah, that makes sense. Interesting. Okay, so continuing. To test whether serological infection criteria represented symptomatic infections, we compared self-reported influenza-like illness in the interval directly preceding the antibody peaks. Indeed, reporting of fever and cough were significantly associated. So what they wanted to see was if you did get infected, uh, was it symptomatic? So were you actually sick from getting reinfected? Or did you just get reinfected and then elicited an immune response just to get rid of the disease, but you didn't show any symptoms? That is something else that can happen. Granted, what it sounds like to me is that a lot of the symptoms of these viruses, especially these coronaviruses, is a result of your immune response acting up in the first place. So I'm starting to like change my mindset to the point where if I'm eliciting an immune response, that immune response in of itself is the pathology of the virus. So yeah, that's sort of what I'm starting to wrap my mind around. The at least the symptoms of the virus are the result of your immune system. That is what I'm starting to come to terms with. Granted, not all of the damage is from your immune system. A lot of the damage is from the virus itself. But what I feel and what symptoms I present to the world is going to be oftentimes my own immune response causing those symptoms as a result of the infection. Continuing, indeed, reporting of fever and cough were significantly associated, and P.031, it's 0.05, and supplementary table S1. So this is, so here's the start and the end. Okay, so this is subject 1, 2, 3, 10. And this is the start and ending years of their testing. And then this is the age when they started and the age when they stopped. These are the months, the number of months that they went in for their follow-ups. And this is the years that those follow-ups covered. And here are the coronavirus infections associated with the antibody increases, associated with the nucleocapsid protein antibodies for these particular strains. And as you can see here, subject 1 caught NL63 4 times over the course of those 22 years. So there's about 5 years in between. He caught 229E every 3 years. Or not, he caught it 3 times, so that would be about once every 7 years. He caught HKU1 once, and so if we scroll down here, 229E seemed to be the most likely virus to be reinfecting people over and over again. Then NL63, then OC43, then HKU1. Okay. Anyways, there was no statistically significant. Oh, median reinfection times of 33, 31, 27, and 46 were found for HCOV, NL63, 229E, OC43, and HKU1, respectively. So here they're saying it takes about 33 months to become reinfected with NL63. Now, that doesn't mean that 33 months is the time frame at which your immune system is then weak enough to be reinfected again. Because what first needs to happen is your immune system has to weaken enough, and then you have to get exposed. So we can safely say that your immune system has become more naive to this NL63 strain before 33 months. So less than 33 months, you are able to be reinfected again by NL63. And we would presume that there is a lower bound to this, and that lower bound is probably, you could probably see that lower bound in this data eventually, or not this particular table, but in a different graph that plots it. You will probably see, okay, what is the minimum amount of time before you could get reinfected? But since these coronavirus strains, they're not everywhere because people have resistance, so they're very slow spreading throughout the world. It could take you a significant amount of time after you become immunocompromised for it to actually be exposed to it, right? And so after you've gotten exposed, so there might be a long period of time where you are actually immunoincompetent, but you just haven't been exposed to the coronavirus, this particular strain of the coronavirus. Continuing. There was no statistically significant difference between the infection interval lengths of the individual virus. Let me read this again. There is no statistically significant difference between the infection interval lengths of the individual viruses. Okay, I mean, this to me seems like there is a difference, but perhaps it's not statistically significant. I would assume between 13 and 49 that is a big difference, but let's keep on going just to see. In a few cases, reinfections occurred as early as 6 months. So this is what I was talking about earlier. There's probably a minimum amount of time where you were just very unlucky. You lost the immunoincompetence, or you gained immunoincompetence in a very short amount of time, and then you unluckily got exposed to someone else who was sick with the same virus and got infected again. So in a few cases, reinfection occurred as early as 6 months, 2 times for HCOV 229E and 1 time for HCOV OC43, and 9 months, so 2 times for HCOV NL63. Interesting. So the most frequent observed reinfection time was 12 months. For reinfections occurring as early as 6 months, we observed no reduction in antibodies between infections. So for reinfections occurring as early as 6 months, we observed no reduction in antibodies between infections. Okay. At longer infection intervals, intermediate reductions in antibody levels were observed. So what they're saying is if the reinfection occurred in that short amount of time, they probably weren't able to see the difference in the antibody levels. So anyways, we observed no reduction in antibodies between infections. At longer infection intervals, intermediate reductions in antibody levels were observed. So you could see the reduction. So I'm trying to understand this then. So how would they know if someone was reinfected if their criteria was to see a 1.4-fold increase in the antibody levels? The only thing I can think of is it starts off high, goes down at the 3-month interval, and then goes back up at the 6-month interval. But yeah, I'll have to look at the data just to verify that. That doesn't jive well with me. Anyways, we observed no reduction in antibodies between infections. At longer infection intervals, intermediate reductions in antibody levels were observed. Anyways, the ability to detect short-term reinfections is limited by the sampling interval. Importantly, though, no reinfection was observed at the first subsequent follow-up visit after a 3-month interval, so no one got reinfected within 3 months, according to their criteria. We did observe several reinfections at subsequent visits with a 6-month interval, suggesting that reinfection within 6 months do not occur. Wait, we did observe several reinfections at subsequent visits with a 6-month interval, suggesting that reinfections within 6 months do not occur. Oh, so I think what they're saying is uh, the reinfection is happening between 6 and 9 months, if I'm understanding that correctly. So the reinfection within 6 months does not occur. Okay, so they're saying it takes more than 6 months, but not much longer after 6 months. Man, I'm having a hard time really following this. Anyways, to further support this point, we examined ratios between antibody levels between 2 visits, both for 3 and 6 month follow-ups. As shown in figure 1b, only ratios below 1 were found in the observations with every 3-month sampling. And we can therefore safely conclude that reinfections occur from 6 months on, yet most probably not earlier. So let's go through this figure. So figure 1, infection and reinfection characteristics, and waning immunity for seasonal coronaviruses. So the interval duration between the reinfections, only reinfections which were observed within a continuous follow-up period are shown. Only which were observed within a continuous follow-up period are shown. So white dots indicate reinfections for which no intermediate decrease in antibody levels could be observed. Black vertical lines describe median reinfection time. So this is for A. So let's look at this real quick. So this is the time frame it takes in between. So for the people who got reinfected, or the times that you could see reinfection, this is how long it took to be reinfected with that particular strain. So as you can see here, now this is interesting. So this is only saying if you did get reinfected, then that is populating this graph. So here you have HKU1, and HKU1 seems to be one that takes a significant amount of time to be reinfected with. Granted, that could be due to the antibodies, or that could be due to the fact that it's just a less pathogenic or a less virulent virus in general, and so it just spreads throughout the world much more slowly. So even after you've lost your immunity, you might still be protected due to herd immunity. You might still be protected for quite some time. So HKU took a long time. So the earliest was, it looks like 30 months. For OC43, you had one case where it was in 6 months, and then 3 cases where it took 12 months, and then you start having more cases where it's only about 2 years. About 2 years later, you get a lot of people who seem to get it again. Okay. And then for 229E, you have 2 people at only 6 months, and then here you just have a lot of people just after a year who get reinfected, right? So one could look at this and say, okay, 6 months is probably where you are vulnerable again, but then around 12 months is when you're actually showing where you've actually become infected again. Interesting. So, and then similar thing for NL63. Anyways, changes in antibody levels post-infection relative to the follow-up interval duration. Each circle represents an infection. The x-axis describes the time until the next follow-up visit post-infection. The y-axis describes the change in antibody levels at the subsequent visit. Larger circles represent a higher ratio rise in antibodies at the initial infection. The horizontal line indicates the border between increases or decreases in antibody levels in the next study visit. Okay, so I'm gonna blow a gasket trying to understand that word vomit. So here's the antibody ratio at the next visit. So one would assume that the next visit you would have less antibodies because you have already caught the infection. So you would hope that the antibody levels would, or you would presume that the antibody levels would generally go down after you've fought off the infection, right? So here they're saying you can see that the levels have decreased significantly, but the size of these, if I'm reading this correctly, so each circle represents the infection, the x-axis describes the time until the next follow-up visit post-infection, and then the y-axis describes the change in antibody levels at the subsequent visit, and larger circles represent a higher ratio rise in antibody levels at the initial infection. So it's sounding like the bigger the circle is, the more their antibodies increased since the previous checkup. Okay, well, okay, that's important, but I'm not going to go over it. Anyways, so 3, the Kaplan-Meier curve showing a decline in antibodies post-infection. So, and the visit at which the infection was established was counted as time point 0. Cases were subsequently followed. An event is defined when the antibody levels drop below the indicated level. When the antibody level did not decrease to the indicated level, an observation was censored at the last time point of an active follow-up. So here they're saying the proportion of elevated viruses. So as time increases, you can see that there is more and more of a depletion in antibodies. And so when people's antibodies had been depleted by 50%, they will be taken out from 100% and it lowers. So here you can see that if your threshold is 50%, so their antibodies get below 50% of the peak, then they are taken out and then the curve drops. And so here you can see at 48 months, pretty much everyone has less than 50% of the antibodies at the peak of their infection. Okay. Now, what I'm not understanding is the 100% depletion. So maybe, I guess that depends on what your definition of elevated antibodies are. So I can't decipher this too much. So I'm just going to continue down, but just know that this line means that there's 100% depletion of their elevated antibodies. So maybe elevated is a certain threshold, and then here it's only 75% depletion, and here it's only 50% depletion. But as you can see, most people experience at least 50% depletion after only 6 months, or at 6 months. Most people are less than 50% of the elevated ratio. Interesting. So, continuing, antibody dynamics after infection. So on some occasions, antibody rises reached high levels, represented in Figure 1b. as large circles, yet these high values were never sustained at the next study visit. To investigate these dynamics further, we calculated the time until 50%, 75%, or full return of antibody levels to baseline, so the pre-infection autoantibody levels, and as can be seen in Figure 1C, within 6 months post-infection, the majority of people lost 50% of their antibodies, whereas after a year, the majority lost 75%. So a complete return to baseline levels occurred within 4 years for half of infections. Oh, okay. So this makes it a lot more sense. So they determined what the baseline it was based on the um, their antibody levels before they were reinfected and the antibody levels while they were infected, which is pretty high. And then they determined these percentages based on the difference between the baseline and the elevated levels. So here you can see, but after 6 months, most people were less than 50% above baseline, which means that they lost most of their antibodies already, or most people have lost most of their antibodies. There were still about 25% of people that had more than 50% higher than normal antibodies. Granted, is it still protective? Probably. It's probably still protective to have that amount, but yeah, and also you want your antibodies to go back down, but you want your baseline to be high enough to still protect you. Like that's the whole point. Is to have a good baseline. And then some people or most people got 100% depletion after only 4 or 5 years. So they went actually even below their previous baseline. Interesting. So good thing that I read that. Anyways, simultaneous infections by alpha and beta coronaviruses. Although our ELISA test using the C-terminal part of the N protein were cautiously designed to be specific for each individual virus, we cannot rule out that a certain degree of antibody cross-reactivity occurred. We therefore investigated how often infections coincided. Since cross-reactivity may lead or may have led to false labeling of infections, we observed no significant simultaneous infection for alpha coronaviruses alongside beta coronaviruses. However, we did see that infections by the beta coronaviruses often coincided. Likewise, for alpha coronaviruses, infections coincided with in 59.5% of cases, and vice versa. Hence, there is a risk that we overestimated the number of infections and thus reinfections. We therefore reanalyze the data with a more stringent definition of infection. Okay, so this is good. So they changed their definition of infection to try to remove the effects of cross-contamination with the other coronaviruses because antibodies can sometimes be cross-reactive, including only the strongest antibody rise induced by a beta coronavirus or alpha coronavirus at a given time point. Under this definition, we still found infection intervals comparable to the original data, with minimum infection intervals as short as 6 months and frequent reinfections at 12 months, although the number of reinfections was obviously reduced. So they did that data analysis again to try to deconvolute, and they tried to use a method that focused on which antibody concentration was a higher peak versus the other, and they still got a similar graph. But I'm curious why they even, I guess they might have wanted to include it just so that people could follow them, because if they just jumped down to this step, a lot of people would be raising their eyebrows, possibly. So the simultaneous infection percentages, and keep in mind simultaneous just means within 3 months of each other, I believe, because they're only following up every 3 months. Interesting. So, broadly acting antibodies recognizing SARS-CoV-2. Okay. In theory, antibodies induced by coronavirus infections may have a broad coronavirus recognizing characteristics. We therefore performed an additional ELISA on all 10 subjects, this time using the complete N protein of SARS-CoV-2. Oh, and to allow detection of broadly reacting antibodies. Visual inspection suggests that broadly recognizing antibodies were produced and were most likely induced by combined infection with HCoV NL3063 and HCoV HKU1. Wait. Let me read this. We therefore performed an additional ELISA on all 10 subjects, this time using the complete N protein of SARS-CoV-2 to allow detection of broadly reacting antibodies. And visual inspection suggested that broadly recognizing antibodies were produced and were most likely induced by the combined infections with HCoV NL63 and HCoV HKU1. So let me see the subjects 2, 9, and 10, supplementary figure S1. Where is the supplementary? Okay, well, I can't find the supplementary figure. Supplementary figure S1. Okay, somewhere there's supposed to be supplementary materials, but I can't find, and keep in mind, this is a preprint paper, so not everything is done. Maybe I'm gonna go on this page and see, okay, so here they have the supplementary material. So I'm gonna click this to look at that real quick. 59 minutes, so. Oh, okay, so supplementary material. Do I need to click it? Okay, good. So the supplementary material was not in the actual preprint. So supplementary material figure S1. Okay, so let's look at this. So this is where they do antibody reactivity of HCoV NL63 and HCoV 229E and OC43 and HKU1 and the full envelope of protein, or N protein, which I believe is one of the envelope proteins of SARS-CoV-2. In red, optical density measured for the C-terminal N protein of HCoV NL63, in yellow, optical density measured for C-terminal N protein of HCoV 229E. Let me. Oh, that was not it. 2, 9, and 10. Okay, so here's subject 2, and this is the cross reactivity. Okay, and then they must have saved their samples or something to do this analysis. So yeah, oh, I see. So maybe they wanted to align the, okay. So it's looking like they used this data to see if there was if one disease might possibly have induced the antibody production of another disease. So between the alpha coronaviruses and between the beta coronaviruses, there might have been some cross reactivity, right? But here, let me just make sure that, yeah, and here they did those ELISA tests, which is an antibody test done on a microwell plate. They did that with the SARS spike coronavirus and saw if there were elevated levels. And it looks like, it looks like here there was an increase, right? And that increase sort of corresponds with OC43 and HKU1. Now let me just see if NL63 and HKU1. Oh, so NL63? Maybe there's a little spike there, but gosh, this is a little bit too much for me. So let's look at subject 2, subject 9. HKU1 and NL43. Okay, maybe between those you have an increase of those, and so therefore, maybe. Let's see, and then 10. So they had an increase here, and maybe it required 2 antibodies to work. Let's just keep on going on. So they basically said there might have been some cross-reactivity between the envelope proteins of the HCOV NL63 and the HCOV HKU1, possibly. Coronavirus infections in changing seasons. To date, it is uncertain whether SARS-CoV-2 will share the same winter prevalence peak that is observed for seasonal coronaviruses in non-equatorial countries. However, it is important to consider that the winter preference of seasonal coronaviruses has only been determined by testing respiratory samples of people that experience disease. So keep in mind, there might be, it might not be that people are actually catching the disease more frequently. It could, during the winter months, it could just be that they're more susceptible to presenting symptoms at that time, and therefore are more likely to be tested. Granted, another thing could also be that the more symptomatic people are, the more virulent or the more virus particles they are exuding into the world. So it is possible that during the winter months, people are both more symptomatic and more people are getting infected as well. So, this could go into why herd immunity isn't always, it's not a fixed number, because if there are periods of time in which the virus is naturally going to infect more people, well, then what is defined as herd immunity is going to be a higher percentage of the population than, say, during the summer months, where more people are likely to be outside, and so there's probably going to be less buildup of the virus particles in whatever environment that they're in. Small things like that can really influence the basic reproductive number, and therefore influence the standard percent of the population that it would be required to have a herd immunity protection. Continuing. Sampling and storage is therefore dictated by having symptoms and not by study protocol. If coronavirus spread continues unabated in summer, yet people rarely display symptoms, possibly due to higher vitamin D levels, which is something that can help regulate the inflammatory and immune response, which if we're assuming or reasoning that a lot of the symptomology is associated with a dysregulated immune system, then having higher vitamin D levels might be very beneficial at helping control that. Continuing, and are therefore not sampled. Infections will remain undetected. However, our serological study is unique because it avoids this sampling bias. So they're working with these 10 people and they're doing regular follow-ups. And because of the regular follow-ups, they are going to be avoiding that sampling bias that we get in hospitals and clinics, because the only people going to hospitals and clinics are people who are or hypochondriacs, right? Or are experiencing more serious disease, because a lot of people get sick, but they don't go to a clinic because why spend that money, right? Anyways, the Netherlands has a typical temperature or temperate climate, and our study samples were collected at regular intervals. The sampling of each subject started at different times of the year, and because of the 3 or 6 month regime of visits, months regime of visits, samples were collected through all seasons. Consequently, we can, for the first time, visualize in an unbiased manner the seasonality of coronavirus infections, as shown in Figure 2. So here they're actually verifying that it is not just a bias in the data or a bias in the severity of disease that is causing the highs and lows of the viral infections, at least for the common human coronaviruses. There seems to be an actual periodicity to them that is independent of, or I shouldn't say independent, but there's a portion that is, in fact, orthogonal or independent to uh, the severity of the disease. So more people actually are getting infected during the winter months. Starting in December, then January, February, and then dropping precipitously once the summer months hit into the late summer. So one could probably argue that during these times, the basic reproductive number of these viruses is very high due to whatever climactic or socio conditions are available. And so a lot of people get sick or contract the virus, and then a lot of people start developing immunity, right? And if a lot of people end up developing immunity, um, and then the summer months hit and the basic reproductive number goes down, then you'll have a period of time in which you are going to be well above herd immunity, because the basic reproductive number of the disease has gone down, which is why even into September, um, you have low incidences of contracting these viruses, and then it goes up again. So everything's a cycle. So here's the discussion. We show for the first time that reinfections with all seasonal coronaviruses occur in nature. The majority of reinfections occurred within 3 years. If SARS-CoV-2 will behave like a seasonal coronavirus in the future, a similar pattern may be expected. A similar pattern will definitely be expected if it ends up adapting in a way that makes it much less virulent and lowers its basic reproductive number to be closer to that of the other coronaviruses. If that ends up happening, then yes, I can definitely see it following a similar pattern. However, I could also see there are some molecular reasonings why this won't happen. So I've discussed this in previous videos, but the SARS-CoV-2 coronavirus, the SARS virus, and the MERS virus, all of their spike proteins, and keep in mind the spike proteins of viruses are trimers of I should say the spikes are trimers of the spike proteins, or the S proteins. And these S proteins are on the surface of the virus. And for SARS-CoV-2, and MERS, they are in a partially open state where the receptor binding domain and the receptor binding motif of the spike protein is a lot of the trimers are partially open. So one of these subunits is sticking out into the world in a pretty active form. And then the other two, or at least one, are closed and they're protected from the environment in some way. With the 4 human coronaviruses that spread out or that are currently endemic to the human population, their trimers are all closed. They're all protected, and I think pretty much all of the studies have it so that they never have them partially open. They will only open up under the condition that they're very close to the receptor. And we assume that this type of protective measure put in place by these normal human coronaviruses is making them much less virulent and therefore making their basic reproductive number lower. Um, but this is not the case of the SARS-CoV-2, SARS and MERS. So for SARS and MERS, the only thing that stopped them was our collective will to stop them and to basically kill these viruses and prevent them from becoming endemic in the human population. With um, with SARS-CoV-2, what my concern is that it does become endemic to the population, and it does not have a good reason to be, it doesn't have a good reason to mutate in a way that makes it less contagious as for now, which would mean that it rather than the typical 3 years, so we can assume safely that after 6 months you then become reinfectable for a lot of these coronaviruses. If it's a similar time frame for SARS-CoV-2, then we would say 6 months, like the others, but rather than taking on average 2.5 years to become exposed to that coronavirus again, it'd probably take a lot less time to get run into someone else who has been infected, right? Now, granted, this has nothing to say whether or not the second time you've been infected, if it's going to be nearly as bad. You might be very, very lowly symptomatic or asymptomatic. But that is currently unknown now. And with, I guess, my research into the antibody-dependent enhancement, it very well could be worse, especially if the antibody levels get down to a non-protective or anti-protective level. Because with antibody-dependent enhancement, it seems to be that if you have protective high levels of antibodies, you are protected from infection. But if you have low levels of antibodies, you are either prone to infection because they're so low that you end up, if they're so low that you end up not even being able to partially protect yourself from them, then they're just going to reinfect you like normal. And then there's probably going to be a sweet spot where you have some antibodies which would permit the virus to get into immune cells very easily through ADE. Granted, we do not know which one, if this is going to be happening for this current coronavirus, but I have very little confidence that it is not happening because of we have all these other viruses that are closely related that are using the same mechanisms to get into cells, and yeah, it's not looking good in that regard. And at some point it becomes a catch-22 because some of the issue is going to be if you do produce the wrong types of antibodies that end up causing autoimmune attack. For one, they're not even going to be as effective at protecting you, right? That is something else that I gotta consider is if the antibodies are also causing an autoimmune attack, then it's going to effectively, for the virus, be a lower concentration of antibodies because a lot of antibodies are going to be consumed with attacking your own body. So that might actually permit a virus to use ADE even though the actual concentrations of antibodies are really high. Because if they're really high, but a lot of them are being used to attack your own body, then it could be perceived as a low level amount of antibodies, which could theoretically cause, especially if they're antibodies that allow the receptor binding domain to be active, then ADE could happen, even with high antibody concentrations. Okay. I think I'm understanding this a little bit. So, continuing. However, this time span between infections does not indicate an individual's protective immunity lasts for the same period of time, as reinfection is also dependent on re-exposure. So this is what I was talking about earlier. It's not just when your immune system has become incompetent again. It is that plus the time of until you are re-exposed. In fact, based on the minimum infection interval and the dynamics of antibody waning that we observed, the protective immunity may last as little as 6 to 12 months. These people modeled the protective immunity and reinfection dynamics of HCOV OC43 and HCOV HKU1, an estimated a 45-week period of protective immunity. 45 weeks. That is less than a year. So our serological study confirms this prediction. When we view our findings in light of the current control actions taken for SARS-CoV-2, it is clear that coronavirus reinfection risk is key to public health policy. Here we reveal a risk that in the near future, serology-based tests that measure previous infections for SARS-CoV-2 may have limited use if that infection has occurred more than a year prior to sampling. Additionally, vaccine studies should anticipate that sustained protective immunity may be uncertain for coronaviruses, and repeated yearly or half-yearly vaccinations may be needed to circumvent ongoing transmission. And this would be such a loss for humanity if we ended up being so, what would the word be? If we needed to have 6-month vaccinations, boosters at all periods of time, that would be such a loss for humanity, because think of all of the manpower and money and just generalized sickness that would be the result of this need. To have this need would be a loss on humanity by far. That would set us back. And I mean, what also worries me is I want to make sure that I'm making the right antibodies. I want to make sure that I'm making antibodies that do not permit antibody-dependent enhancement. I want to make sure that I'm not making antibodies that cause autoimmune attack. Hell, probably part of the reason why the antibody levels for these coronaviruses seem to drop so quickly, or more quickly compared to other viruses, is that these antibodies, a lot of them might actually be causing autoimmune diseases. So we really need to be meticulous in our treatment design. Continuing, there is ongoing discussion regarding herd immunity for SARS-CoV-2 control, and herd immunity occurs when a threshold proportion of a population is immune to a certain pathogen and protects even non-immune individuals against the infection by limiting overall spread. And this spread has been observed in a variety of viruses, mainly those that are partially controlled through vaccination programs such as hepatitis A, influenza A, and human papillomavirus. In the case of SARS-CoV-2, achieving herd immunity may be challenging due to rapid loss of protective immunity. Granted, it would be nice if they had some references as far as what are the rates of loss of protective immunity for these other diseases. That would be really good to know. I mean, for influenza, it's not that long. Granted, there is semi-protective immunity, so even if you do get vaccinated, even for the wrong strains of influenza, there's oftentimes enough cross-reactivity to confer some protective benefit to the host. Anyways, it was recently recognized that recovered individuals should receive so-called immunity passports, and I freaking hate this idea, but because it could encourage people to go and get the disease in the first place, right? So I'm not liking this sort of authoritarian measure being put in place, which would allow them to relax social distancing measures and provide governments with data on herd immunity levels in the population. However, as protective immunity may be lost by 6 months post-infection, the prospect of reaching functional herd immunity by natural infection seems very unlikely. Very unlikely. So we should take lessons from these closely related coronaviruses, and we should use the precautionary principle when discussing this current pandemic with this current coronavirus, because the protective immunity might only last for 6 months. And I don't want to be one of the people serving as a guinea pig for the first reinfection case. I don't want to be that person. I want us to have the disease spread slow enough that we end up being able to collect data about what happens when you get inoculated a second time, and a second time after a few months, especially. So it is not a question of if, it is a question of when. Because with all viruses, you will get reinfected, or I shouldn't say you will, but you are susceptible to being reinfected. That is true. What happens when you're reinfected completely is determined by what your immune response is. Is your immune response sufficient, and is it accurate, and is it precise in its ability to protect you? If that is the case, even long after your antibody levels get below certain concentrations, if that is the case, then we are safely protected and we will end up reaching herd immunity, especially after we get vaccinations. What does not bode well for me is what if after 6 months the first person we end up discovering one of the early people who got infected once, verified through a polymerase chain reaction or PCR test, if it gets verified that they become infected again and it's worse the second time around, that is going to be a big problem. In which the only recourse of action would be to have everyone prepare to completely 100% socially isolate themselves for 3 months worldwide. Like, that to me seems to be the only course of action for stopping this. So what we would probably do is have quite a few months of preparation to make sure that everyone has enough food in their homes and everyone has enough medicine in their local communities, so they wouldn't have to travel to be healed. And then we would find ways to automate as much as we could during that time, and then we would just shut down. That is my guess. If that ends up happening, where we find out that antibody dependent enhancement is causing an increase in severity of disease at the second time of infection, what we will end up doing is shutting everything down. And the only things that will continue running are jobs that you can safely self-isolate while doing. So things like farming, gardening, um, certain types of manufacturing. If you are able to do it in a socially distant manner, uh, we can make things work. We can keep the economy running, and we can keep on surviving through all of this. But it really is going to take a lot of collective will across the entire world to do that. Because and granted, this is all very speculative. This is all very speculative, but based on what I'm reading regarding related coronaviruses, regarding the mechanisms by which coronaviruses are able to get into cells, and the mechanisms by which viruses are able to suppress immunity and confuse the immune system, um, and the mechanisms by which this coronavirus makes it much more virulent than other coronaviruses. All of these speak to me in a way that if I put all the puzzle pieces together, that might be our only course of action in the coming months. Or I shouldn't say that, not our only course of action, but that is going to be the result of all of our hardship. that is probably going to be the result of our hardship, is that we will find that this has become endemic. And it's not just endemic in the sense that herd immunity always lowers because of people being birthed. It might be endemic in that herd immunity is a farce. It does not last, and it will continue to be a problem um, for our bodies to fight. So um, I'm not looking forward to that. I'm not looking forward to the next few months. I'm the only indicators that I see against that is that the convalescent plasma therapies seem to be very beneficial. Granted, that's coming out of people who have recently recovered from coronavirus, so we presume that their antibody concentrations are pretty high, right? We cannot say the same for 6 months down the road, right? What an absolute what an absolute loss. You know, I tried warning a lot of people about how serious this was, and um, I really hope that there's something different about this virus that doesn't let these puzzle pieces fit together the way that they are. I really hope um, that there's just something different about this virus um, that does not have it follow the pattern of the other coronaviruses, so that it is no longer that it is no longer endemic. I'm really hoping that that's the case. We're gonna see. We're gonna see how long our collective humanity and our collective will lasts, because it seems to be very fragile right now. Anyways, that is way too much musing. Continuing. So it is generally assumed that exposure to seasonal coronavirus does not equal the beta coronavirus. HKU1 is considered to have the lowest prevalence, and we found the same. We also observed that antibody levels of HKU1 were low over the whole range, over all samples tested. Whether this indicates that HKU1 infection elicits a poor induction of antibodies is intriguing, yet not answerable in our study. So I would assume that um, probably one of the reasons why you don't need to produce a lot of antibodies is that it might be a very uninfectious virus or virion, and therefore you don't even need that very many antibodies to render it useless. And you could argue that it's not very infectious because it has the lowest prevalence in society. So the more infectious it is, uh, you would probably you'd probably be able to duplicate more within the host. But if you only need very few antibodies to get it under control and to get the for lack of a better term, the reproductive number of the virus inside of you low, then you probably don't need to elicit a lot of antibodies, which might be good because having too many antibodies could be damaging for your own tissues. We used a relatively small part of the N protein as the antigen in our test to most specifically identify infections by each virus individually. This small part of protein may not contain the most immunodominant epitopes of HKU1. So as we said, we used a portion of the virus protein, so not even the whole protein, just a part of the protein, to do these antibody studies. It could be that for this particular strain of HKU1, that the part of the protein that they used was not a very important antibody that the body produces. Because remember, your body actually will produce many, they'll produce polyclonal antibodies for every disease that you run across. You're not just going to attack one part of a virus, you're going to attack as many parts as possible. And they're saying here that this might be, may not contain the most immunodominant epitope. So it might be that you're fighting the virus with different antibodies, and because you have these other antibodies in high concentrations, you might not need the antibody that attacks this small part of the N protein of the HKU1. Anyways, neutralization tests would ideally be done to solve the issue of protective immunity raised by HKU1. However, unfortunately, no culturing system suitable for neutralization testing is available for this particular strain. So that probably is the result of there probably isn't a good cell line that is able to take this up and culture it very well. So yeah, that's probably the reason why that study hasn't been done. We noticed three subjects to carry antibodies recognizing SARS-CoV-2 N proteins at certain time points. It is unlikely that they had been infected with SARS-CoV-2-like virus in 1985. So, and we therefore suggest that broadly acting antibodies may have been induced by coinciding infections of alpha and beta coronaviruses. So in our subjects, it's HKU1 and NL63. So they're saying here that when they take the SARS-CoV-2 N protein and they run it through the same tests using the old blood samples, it looks like they are finding antibodies against SARS-CoV-2. But that's obviously SARS-CoV-2 did not exist at the time. So what has to be happening is some different antibodies are being produced that are able to cross-react. And they're making the argument that maybe being infected by two at the same time, maybe the antibodies they produce are each one very weakly able to attack SARS-CoV-2, but when you have both of them at the same time, the antibodies might be able to combine their forces in a way that generates a signal, or generates a significant signal. So this could tell you more, in that it could tell you that sometimes there are these antibody studies that we're doing on people to see how many of them have contracted SARS-CoV-2. It might be very difficult to deconvolute the prevalence of these other coronaviruses that are spreading around. For instance, I mean, think of New York, highly populated and highly concentrated. Using their public transport systems, subways, all that. It's a very good vector, not just for coronavirus or not just SARS-CoV-2 to spread, but all the other diseases, all these other coronaviruses to spread. So if we're saying, if the likelihood of having cross-reactive antibodies can be determined by having antibodies, high levels of antibodies for both an alpha and a beta coronavirus, then in the city, you're more likely to contract each of them, and therefore you're more likely to contract both of them, right? So what might be the case in some areas of the world is that if you're in a highly populated area, a densely populated area, your antibody study might be poisoned by the fact that these people are actually contracting both alpha and beta coronaviruses of the common human coronavirus, and it's completely independent of the SARS-CoV-2 protein. So what this goes to show you is, with antibody studies, it is so hard to know if you're doing good science. It's so hard to know if you're doing any of this correctly. And yeah, so I'll just keep on going. I'm getting too depressed. To explore this finding, we looked at the genetic distance and consequently amino acid differences in the structural proteins of the various coronaviruses, notably the SARS-CoV-2 N protein, and only has 32 and 34% identity to the amino acid level of these two coronaviruses, and only 26 and 24 for these two. Similarly, and they split them based on the alpha and the beta coronaviruses. Excuse me. Similarly, the distance between the alpha coronavirus and beta coronavirus end protein is large, only 24 to 26% amino acid identity. Still, we cannot exclude the presence of conserved or conformational epitopes in these. So there are ways that you can change a lot of amino acid residues on the protein. So remember, proteins are made out of amino acids, and by changing the amino acids, you can change the structures of proteins. But there are many different. It could just be that only a few amino acids are doing the majority of the work as far as what antibodies are being produced against that section of the protein. They're saying that might be, even though there's low sequence homology between these coronaviruses, there could be some very specific conserved residues, or there could be very specific conserved residues where they're not the same residue, but they might be a very chemically similar residue. Or it could be the shape, because the shape of that particular protein might be conserved, even though the amino acids change. So they might have a completely different combination of amino acids that confer the exact same shape, and therefore you could have some antibodies crossing over between them. It's all hand wavy right now because I don't like all of this is just biology is noisy. That is the takeaway from all of this, is that biology is is a really noisy field of study, and it's very difficult to remove confounding factors from each other. Continuing more broadly, acting antibody response due to simultaneous exposure and concurrent infections. Additional screening, including more subjects, is required for confirmation. Our study was subject to limitations. One is the inability to sequence the virus genome during infection. In theory, strain variation could play a role in susceptibility to reinfection. So it could be the viruses themselves are mutating enough to make someone susceptible to being reinfected. So it might have nothing to do with the antibody concentrations at all. And so it would be nice, it would have been really nice if they were able to get the genome sequence or the sequence of the proteins to do these experiments as well. But alas, we didn't have that technology 30 years ago. So that really would help things a lot. You know, granted, if we had genomic technology when the Spanish flu was hitting, that would really elucidate a lot of our open-ended questions on why was the Spanish flu so bad. Well, if we could dig into the genomic information and see how the genome changed over time and location, that could have told us a lot about the severity or why it was such a severe disease. Anyways, continuing, HCOV NL63 and HCOV O43 and HCOV HKU all show different co-circulating genetic clusters. For 229E, there are no major genetic subtypes known, like the current SARS-CoV-2 situation. It could be that our observation of short-term immunity is influenced by genotype variations, and a study on protective immunity would therefore ideally allow sequencing of reinfecting strains from respiratory material. However, this is intractable and in a natural infection study because the virus shedding in reinfection can be as short as 1 day. That's annoying. So to do these studies, keep in mind they have all their blood samples, right? They have their blood samples. But if you want to do genetic studies on the coronaviruses, you would have to use a sample that had a high enough concentration of the viral genetic information. So for a lot of these common cold coronaviruses, they don't have a significant level of viremia, so virus in the blood, viremia. And if they did, they probably would have been able to go back into the blood samples and do these genetic studies on the virus strains to see which ones caused, see if there was significant genetic variation causing the reinfection rate. But that's not the case. And in order to do that, you would need to be doing nasopharyngeal swabs, or you'd need to be doing some sort of swabbing to get enough viral genetic information. And unfortunately, with some of these coronaviruses, it only they are only there's only significant viral shedding in for 1 day, right? So you just have to be lucky enough to get it on that exact day. That's not very possible. Anyways, so but at least with SARS-CoV-2, there probably is significant viremia. So in the future, we could be doing studies where we take blood samples from people periodically and do this exact same research with people. And we could theoretically go back on the blood samples and test for viral genomic DNA or RNA. Anyways, another limitation is that the studies subjects were all male. For COVID-19 and also HCoV-NL63, men have a higher incidence of disease and is therefore of interest to determine the dynamics of protective immunity also in a cohort of healthy women. So yeah, that we know that men disproportionately show higher rates of complications, right? So maybe with women, their antibody levels might be spiking all the time, right? And that might be conferring protection for them, and why they're not as sick from these diseases as much or as frequently. Who knows? It'd be nice if 30 years ago they had that foresight to have a cohort of women as well, but they simply didn't. Anyways, in conclusion, seasonal human coronaviruses have little in common apart from causing common cold. Still, they all seem to induce a short-lasting immunity with rapid loss of antibodies. This may well be a general denominator for human coronaviruses. So, on that very grim note, hopefully the peer review process will highlight some issues that would completely disagree with this, and by highlighting those differences, they end up saying, yeah, we shouldn't even believe that it is likely to be reinfected by SARS-CoV-2. But I currently have no, what would the word be? No confidence in that. So anyways, with that, if you have any questions, please leave them in the comments, and I will see you tomorrow. Hopefully I will find a better paper to that is a little bit more uplifting than this. So have a good one.